very much, Tim, and good afternoon, everyone. And thanks for the opportunity to listen to another talk. Appreciate Pastor John William Noble and everyone who's organised today's event, the chance to once again bring a heavy topic to us, but one that I think is important for the church to consider, which is the topic of the safety of the COVID-19 vaccines and how we got to where we are today. So in today's talk, I'd like to consider four key areas. The first one is going back to first principles, going back to the clinical trials that preceded the rollout of the vaccine. And I'd like to look at 5 case studies of actual harm people suffered during the time when the COVID-19 vaccines were being trialed and tested. Secondly, I'd like to take a sort of high-level, 1,000-foot view of public health trends over the last few years to review and see whether, since the rollout of the vaccines, we've become healthier worldwide in global public health, or whether we have become sicker, whether there are any potential signals. So we'll look across the private health insurance industry, we'll look across other researchers across the world, we'll look at several different nations and key research coming out to answer that question. Thirdly, we'll then go from the very high level to the very microscopic, and we'll look at a few detailed areas. In particular, you may have heard of some of these clots, sometimes known as calamari clots, that have been forming in people and embalmers and undertakers, and funeral directors say they haven't seen them of this type before. They have a different consistency, they have a different texture, different size to what's been seen before. We'll also take a look at contamination, very key theme morally as well. We'll also look briefly at IgG4 antibodies and some potential immune risks from that. And then finally, we'll consider views of the vaccine within the church, the world, and elsewhere. And we'll try to tie it all together by using the Word of God. So before we get into the clinical trial case studies of harm, do you remember some of the headlines announcing the dawn of hope that the vaccines bring us? Light at the end of the tunnel. 2 giant leaps to freedom. What a shot in the arm for Britain. Hope in the post for 5 million. On its way. Vaccines for over 70s start today. The scientists have done it. Hope at last. A great day for humanity. 1 small jab for Maggie, 1 giant leap for all of us. It's kind of a mixture between the messianic and the moon landings, I think. But here's the reality. Our first story of real harm, actually this young girl was from a Christian family. Maddie de Garay in America. She was only 12 years old when she was volunteered along with her family for the adolescent COVID trial for the Pfizer-BioNTech mRNA shot. And you can see in those pictures at the bottom, she's not looking too well now. But she used to be a very healthy young girl, very active. They would only have enrolled those who were pretty healthy into this trial at this point. So this is actually a quote from Maddie. At the bottom right, that's her giving evidence with her mum, Stephanie, a Christian family, at the Senator Ron Johnson's round table in June 2021. And Maddie says, in fact, this is her mother actually, upon receiving the second shot, which was the 20th of January 2021, Maddie immediately felt pain at the injection site. Immediately. Over the next 24 hours, she developed severe abdominal and chest pains, and the way she described the chest pain, and I quote, "It feels like my heart is being ripped out through my neck." She had painful electrical shocks down her neck and spine that forced her to walk hunched over. Perfectly active, very healthy, now wheelchair bound, severe problems, nasogastric tube to take food in, so abdominal, gastrointestinal problems. They put it down in the trial as functional abdominal pain. When Pfizer announced their results March 2021 on this trial, you won't be able to see it, but under the headline it says that their product was well tolerated. And the headline says they announced positive top-line results of their trial for adolescents. And when they wrote it up in the New England Journal of Medicine, again it says that there were no serious adverse events that were vaccine-related. So there's the reality, and that's how they spun it, Pfizer. Our second story, going back to the adult trial in 2020, Augusto Ru, a lawyer from Argentina, a young man who's about 35 years of age, After his vaccine, he's admitted to hospital with fever, breathlessness, tachycardia, so fast heart rate, and chest pain. He had to sue to get his medical and trial records. He had a CT scan, and you can see it there, and it showed laminar pericardial effusion. That's pericarditis, that's inflammation of the fluid-filled sac, the pericardium around the heart. And again, same story in the clinical trial. They don't put down his cardiac problem. It just has this idea that he has some COVID tests and also that he had some psychological or mental health challenges, but it doesn't talk about the pericarditis. But on the right, we don't only have the CT scan, we have his doctor, that's Dr. Distilio, his discharge note, and it records adverse reaction to coronavirus vaccine, high probability. It's in Spanish. So again, you've got the reality, you've got the way that they write it up. Even if it's, everything is probabilistic, high probability, good chance, you're supposed to write it up and take it a cautionary approach to it as the pharmaceuticals. That's the whole point of a clinical trial. Another one, this is Moderna now, Olivia Tessiner, participant in Moderna's clinical trial, and she took her first dose in August 2020, the second dose in late September. She experiences severe pain in the arm where she receives the vaccine, fever and fatigue. Again, you can see at the bottom she was healthy before, afterwards not healthy. She gets neurological symptoms, severe headaches. This developed 4 days after a second dose, requires multiple shoulder surgeries and lymph nodes under the vaccinated arm so severely disfigured they had to be surgically removed. So what happens? Well, sadly, she ends up developing cancer of the lymphatic system, T-cell lymphoma, and it then develops into full-blown blood cancer, which is leukemia, and she goes through chemotherapy. Same pattern. Dr. DeMarco's referral notes for Olivia Tessiner, I've got them highlighted, confirmed diagnosis of vaccine adverse event. So once again, we have the doctor's own notes that have been released by these people. In Augusto's case, he had to sue to get them, and here we have Olivia's one as well. Brianne Dressen, same story, this time in the US trial for AstraZeneca. Again, super fit, super healthy young woman. She likes rock climbing, skiing, all-day hiking, super fit. Again, a devastating injury in the Utah trial back in November 2020. And remember, AstraZeneca wasn't even approved in America. So when they told you how great AstraZeneca is, it didn't even get the US approval. She was part of the US trial. She experiences double vision, chronic nausea, brain fog, profound weakness, severe neurological problems, suffers from 20 symptoms, including autoimmune nerve damage, chronic demyelinating polyneuropathy. This is severe pain and severe sensitivity to all kinds of stimuli. So sensitive was Brianne Dressen that she couldn't even tolerate light, sound, touch. Even conversations, talking to her own children could be painful. That's the level of sensitivity she faced. Accrued medical bills of hundreds of thousands of dollars, so the pharmaceuticals didn't cover it. Ordeal sadly left her so upset and so traumatized she was contemplating suicide. And she said, similar to Maddie Degaray, I feel like I'm being electrocuted from head to toe. And she also says, within an hour, I developed tingling in my injection arm, which moved to my other arm, my legs and head. I developed this horrific electrical pulsing sensation throughout my body 24/7. So again, you see how short the time is between injection and pain, and you see the severity of the symptoms. And one more, a young man from Brazil. This time, he's also getting the AstraZeneca shot, Clayton Luis de Oliveira Souza. Again, he does have underlying diabetes, but apart from that, he's a pretty fit young man. I've seen him doing like a handstand when he was all healthy. This is a fit young man. He was a fitness fan. He develops epilepsy after his second dose of AstraZeneca in November 2020. He also develops encephalitis, which is inflammation of the brain, and he also suffered a seizure. So he's now on lifelong anti-epileptic medication. And here's a quote from Clayton. My family took me to the hospital. I was released after an overnight stay, went home, had breakfast, went to bed. I collapsed in my bedroom. My family took me back to hospital. I woke up 3 weeks later, having been sedated all that time, spent 2 months in hospitals. Once again, doctors concluded the vaccine must have caused my injuries. So the doctors are actually seeing the patients. They know the score. It's just not being reported by the pharmaceuticals. That's the problem. So the second section I'd like to consider then, we've looked at the very kind of specific stories, actual accounts, actual doctors' release notes. Let's now consider high level, some of the trends in public health across the world, starting with America. 2 reporting systems are on the screen there for vaccine injury. The first one is called vSafe. So in America, about 10.1 million people are registered with the system. It's like an app on your phone, and you volunteer into the scheme. And if you suffer any problems with your vaccines, you can actually report it into the application. Okay, so you actually know how many people have signed up for it. You have 10.1 million, and they all get the chance to have the COVID vaccine. And 782,000 of them, which is 7.7%, actually self-report a health event requiring medical attention following a COVID vaccine. So that gives you a really good rough estimate for how many people are suffering a medical problem or a medical emergency, about 7.7%. And on the right-hand side, you've now got a passive reporting system, the Vaccine Adverse Event Reporting System, VAERS, very similar to our system in the UK, which is called the Yellow Card, run by MHRA. You can see that in 2021, there were 21,000 mortality statistics for COVID-19 vaccines and 526 for the others. And in 2022, 9,102. And for the others, 368. Now, if you look on the screen, you'll see some bars. And you probably can't see any bars apart from those huge red bars which relate to the mortality from the COVID-19 vaccines in 2021 and 2022. In other words, the number of people who self-reported into this system, and anyone can do it, their doctor or themselves, is massive when it comes to the COVID vaccine. We've never seen numbers like it. So again, this screams out that there's definitely something going on and definitely something to be concerned about. And another place we can look for evidence at the very high level would be health insurance, life insurance. People have private insurance, and this is a bottom line industry at the end of the day. If they're paying out more money because more people are getting sick or dying, then they're going to have to talk about it because they're going to have to be accountable to their board, for example. Why is your profit going down? Because you're paying out more money. Well, in Germany, there is a health insurance company called BKK Provita, and one of the directors took the extraordinary step of publishing an open letter in a big German newspaper called Die Welt, February 2022. He looked at his own company's data, health insurance data, and found that 4 to 5% of vaccinees in his company were registered for them requiring medical treatment due to side effects. That's very similar to the 7.7% we just saw from the US VSafe system. Extrapolating this, so saying, imagine if we scale this up to the whole population of Germany. Well, that would be about 2.5 to 3 million Germans would have received medical treatment for side effects. Now, that's about 10 times higher than what the Paul Ehrlich Institute estimated. They said about 250,000, but even they said a pretty big number. But he's actually not doing statistical modeling as such. He's just saying, look, if we scale up the number of people we have registered with my health insurance to the whole population of Germany, you'll get 2.5 to 3 million people. And they weren't the only one, because the largest German health insurer, Techniker Krankenkasse, also reported increase of 40% in claims amongst 18 to 49-year-olds, your traditional healthy population, working age population. 1 in 25 clients of theirs underwent medical treatment in 2021 for COVID vaccine side effects. Quick bit of maths gives you about 4%. So you've got 4%, 4 to 5%, 7.7%. You're getting the idea. This is a pretty consistent figure for how many people actually require medical intervention. You've also seen 5 examples from the clinical trials of people who have serious and life-changing injuries, and it's not that rare. Switching country then, let's go to Japan. This is an interesting story. I'll take a minute to explain this paper. And this is on excess cancer mortality in Japan. So there was a paper, I will read out the title because it's important, Increased Age-Adjusted Cancer Mortality After the Third mRNA Lipid Nanoparticle Vaccine Dose During the COVID-19 Pandemic in Japan. So it will be hard to see all the bars, but I'll do my best to describe them and what's going on. The red bars, which you might be able to see, represent increases in 2022 in excess mortality compared to the historical average over the last few years. And I think in pink color, you'll see 2021. And as you look to the right, you're seeing a few examples of these cancers increasing. This would include ovarian, I think it's pancreatic, and leukemia as well, so blood cancer. Now, and also for the overall, which is the far left bar as well, you'll see a big shoot of red as well. What they're finding is that cancer mortality has increased for several different cancer classes in Japan, and it starts in 2021 and then increases again into 2022, and it jumps both times. And they found, using a technique called prediction intervals, which is similar to confidence intervals, they found that there's a significant jump in several different cancer categories in 2021. I mean, it wasn't there in 2020, but it is there in 2021, and it actually gets worse in 2022. So that's why they've labeled a few of them as being statistically significant. Because you do not expect with either a 95% or 99% confidence or prediction for it to have such high numbers. It doesn't make sense. It's abnormal, statistically abnormal. That's what they found. But the paper got retracted. And it's a very interesting story why it got retracted. Because the paper got attacked for its conclusions. I may not read the whole thing out, but I'll read out as much as I can. Statistical significant increases in age-adjusted mortality rate of all cancers, firstly, and then some specific cancers. So the left side was all cancers, and then the right side, specific types. And they've called out some of them: ovarian, leukemia, prostate, lip, oral, pharyngeal, pancreatic, and breast cancers were observed in 2022 after 2/3 of the Japanese population had received the third or later dose of the COVID vaccine. And in yellow, you can see me highlight, maybe attributable. Maybe attributable. And also, the significance of this possibility warrants further studies. They have looked at the data, seen the jump, done the statistical analysis, and said, hey, it's interesting that it starts in 21, increases in 22. By the way, we had a massive population-wide exercise with the vaccine. Could it be attributable? They didn't say definitely, they said it could be. But that's what they got attacked for. Here's the retraction notice from the journal Curious. The editors-in-chief have retracted this article. Upon post-publication review, it's been determined that the correlation between mortality rates and vaccination status cannot be proven with the data presented in this article. As this invalidates the conclusions of the article, decision has been made to retract. I read the entire conclusion now. That's the entire conclusion. It's only one paragraph in the last slide. At the bottom, just in case anyone's forgotten, may be attributable to, possibility warrants further study. The only thing I can see they can really attack is the title of the paper. But you're allowed to make an observation that it happened after the third dose. In 2022, that's when it really started to increase. So that's the state of academia. If we think that academia is going to save us from all this, I don't think so, not the peer review process anyway. And another expert from Japan, Dr. Yasufumi Murakami, professor at Tokyo University of Science, serves as vice director at the Research Center for RNA Science, holds a Doctor of Pharmaceutical Science at the University of Tokyo, authors over 100 scientific publications. He managed to analyze a database of COVID vaccine recipients of over 18 million Japanese citizens. And what did he do? Just did some statistical analysis. And he was able to conclude the more doses you get, the sooner you're likely to die within a shorter period. A significant peak forms at 3 or 4 months. It's probably due to the vaccine's influence with adverse reactions occurring leading to death. It's a bit grainy, but I have a picture taken from a video still of the paper that he does. You'll see in blue at the top, which is wavy, that's the death amongst vaccinated people over time relative to their last shot. And what you see is that it forms a highly irregular pattern. It's not happening randomly. It's like after a few months, it starts to increase the number of deaths after the last dose. And then it drops down, then it goes kind of random again and goes normal. And in red, the unvaccinated, I think he normalized this to the day that the vaccine rollout got started in Japan, because obviously they don't have a vaccine date. But you see that unvaccinated people will just die, any kind of day. There's not even a lot of seasonality in their deaths. But there's a really irregular pattern with the vaccinated that kind of rises and then falls again. So that's what he's pointing to. He just did statistical analysis, 18 million people on a database, and he knew who's vaccinated and who's not vaccinated. So again, this is just statistical analysis, but people are being attacked from it. I don't have it on the slide, but the US health insurance data, there's even more coming out there. There was 2 health insurance companies and life insurance that pointed to rises in 2021. One group called One America, the CEO said in 2021 that death rates amongst working age people aged 18 to 64 were up 40% in 2021 in the third and fourth quarters over pre-pandemic levels, 40% increase. He also pointed to an uptick in disability claims, short-term and then long-term. That's one health insurance company in America. Second one, Lincoln National, they paid out, not the number of deaths, but the payout for claims for group life insurance deaths, so people dying, again, same age group, 18 to 64, they were paying out 163% more. So again, massively statistically significant, all compared to the previous years. They said that in 2020, that increase in deaths amongst the working age went up only 9%. But 2020 to 2021, it went up 163%. Massive difference. They also point to Lincoln National, the increase in short-term and long-term disability claims. Exactly the same story. Now, just briefly, you might think, well, you can't build an argument based off of just, trends in public health because anything could have driven it, right? Well, if you go back a few years, we did have a drug called Vioxx, a non-steroidal anti-inflammatory drug that was treating inflammation and pain, released by Merck and approved by FDA in May 1999. I would say there's a lot of parallels here to what we've just looked at. In January 1999, Merck launched a large study, but it was found they'd withheld critical data on the cardiovascular toxicity of Merck's drug Vioxx. And in late 1999, Merck's own Data and Safety Monitoring Board identified the risk of a serious heart problems double that of a competitor drug, naproxen. So there was data there in the clinical trials. Didn't we just see that there were actual real examples in the clinical trial, but they weren't spoken of when they released their results? We've seen doctors' notes confirming it, the discharge notes. I've shown you the actual discharge notes. Well, it's just like Merck. They knew they had a problem. There was an epidemiologist whistleblower. He was called Dr. David Graham. Believe it or not, he went through the same whistleblowing channel that Edward Snowden used, like military whistleblowers. That's how tough it is to whistleblow on the pharmaceutical industry. It's not a joke. It's not a small thing. It's a big deal. And he conducted a study. He was just using the public data. He wasn't going back to the trial, just like we did. He just looked at the trends in heart disease and heart-related deaths, cardiovascular deaths. And he was able to project, just using statistics, just like what we just saw, 88,000 to 139,000 heart attacks with a 30 to 40% fatality rate. That was the uptick. Just looking at something must have caused this uptick, something must have happened at the population level. It can't just be climate change, and it can't just be people are stressed by the news. It's got to be something big. Vioxx starts getting rolled out. Maybe that's it. Probably 30 to 55,000 deaths. But FDA tried to suppress him, they tried to destroy him, he goes to the whistleblower channels, and he describes it as the worst public health disaster in history, and his results are accepted by everybody, and they're in the Lancet, they're published. Nobody disputes them. Everybody agrees that Vioxx was a scandal and it's been pulled from the market. So if it can happen there, and that was only 25 years ago, why is it not true anymore for what we've just looked at? And here's some numbers behind, here's a new word for you, iatrogenic harm. Harm from medicine, harm from medical care. Iatrogenic deaths can be caused by a doctor or surgeon's actions. Some examples, medication errors, drug or overdose or dose-related issues. I've heard many examples of people in hospital saying, yeah, they got the wrong decimal point and they got the wrong medication. It happens much more often than you'd think. So always have your patient advocate and always look over your notes because things can go wrong. Diagnostic errors, hospital-acquired infections, adverse events from treatments, insufficient patient follow-up. Again, I know of this with people I know where they're on medication, they don't follow up, their salt levels drop or whatever, and they get sick. It can happen. In 2016, the BMJ published research by Martin McCary and Michael Daniel from Johns Hopkins University. They used previous studies to find medical error at a mean rate of 251,000 a year in America, 9.5% of all deaths. Medical error, third leading cause. Very similar study back in 2000, medical error found to be third leading cause of deaths, accounting for about 225,000 deaths, but it could be as high as 230,000, 284,000 in America. So again, this is all accepted. Nobody disputes this. It's in the BMJ, it's in peer-reviewed journals, it's completely accepted, third leading cause of death. So again, it's not a small phenomenon we're looking at. Well, we've looked at case studies of harm from clinical trial. We've looked at big, high-level trends. Let's take a look now at the very small level, at the micro level. First issue, in the mRNA vaccines, Pfizer and Moderna, at DNA contamination. There's supposed to be RNA shots, but there's been some independent research from some very clever people, including Kevin McKernan, and they found hundreds of billions of plasmid DNA molecules per dose in the shot. And what this is in order to replicate the DNA many, many times, you have to use a vector. They use E. coli. You might know that E. coli, as a bacteria, can be very nasty, okay? It can stop you from swimming in the Thames sometimes, okay? Or in your local beach, right? Because we have to monitor the E. coli levels. But in any case, they use it because, it's a cheap way, basically, cheap and nasty way of making lots and lots of copies of DNA, which you then convert to RNA and then put them into the shots. And by the way, the only reason is to save money, okay? Because they're trying to make profit, not make you healthy. They have to be answerable to their board. That's what counts for them, okay? It doesn't matter how altruistic they might be. If they're not making money for their shareholders, they're not going to exist much longer. And they use, in this study from McKernan, use many studies to detect DNA, qPCR, fluorometry, electrophilic sizing, phoretic sizing, next generation sequencing. And basically, not only finds massive amounts of contamination that's not supposed to be there, much higher than what the regulators said could be there, but also finds there's something there that even the public documentation doesn't say, which is there's something called a promoter called SV40. It comes from the SV40 virus, and it will drive the DNA into the nucleus, which could cause problems to your chromosomes, to your genes. It could even potentially cause issues with chronic inflammation, could have an interferon response. You could even potentially contribute to cancer, because there's a pathway that's been kind of getting more familiar in the last 10 years called cGAS-STING, and it's possible that you can have this chronic state of inflammation, and it can lead to cancer when you overwhelm this pathway that's actually supposed to help you stop getting cancer. So the problem here is bad manufacturing, lots of contamination. It's not supposed to be there, and it wasn't. The SV40 promoter wasn't disclosed to the regulators. And we've shown you the harm we've looked at from the clinical trials. But believe it or not, the clinical trials used a cleaner method. They didn't use E. coli to make lots of copies of DNA. They used a PCR-derived DNA template, heating and cooling, just like a PCR test, to make lots and lots of copies of the DNA. And that's actually much cleaner. They clean it up with magnetic beads. You don't get as much nasty stuff going on, like DNA contamination. You shouldn't have as much of it. And the conclusion here is simple. The safety profile in the trial doesn't really apply to the real-world rollout because it's a more dangerous process. You're going to have more DNA, potentially even bacterial contamination, endotoxin from the E. coli. So if what we saw from the clinical trials looked bad for Pfizer and Moderna, we saw a few studies from them, case studies of harm, it's probably even worse in the real world because the manufacturing process is more dirty. And as mentioned before, the SV40 promoter, forgotten or hidden, I've asked the question, it has, as I mentioned, that nuclear localization signal. So it's going to drive DNA into nucleus. You're going to have foreign DNA probably going to be driven into the nucleus, and that could cause issues, genetic problems to the DNA potentially. So it increases the risk of genomic integration. And it also has with it something called an origin of replication. So it might even make more copies within your own cells as well, of not just the spike protein with the RNA, but even DNA that's not supposed to be there, that should have been cleaned up entirely. And the Epoch Times reported on this, that BioNTech, who partnered with Pfizer, they were really the manufacturers, didn't actually highlight the inclusion of this SV40 promoter. They said because it was a non-functional part of their plasmid, which is what they use to manufacture it, and they've since clarified the information in response to questions raised by the EMA. What they're really saying, and they're trying to play it down, is they wasn't shown to them through the documentation. So this is something that was kind of hidden till one researcher basically found it. So there's stuff in the vaccine that even the regulators probably weren't really told about. And then I've shown this slide before, and it is very important. Again, a little complicated, so I'll try not to go into too much detail. But switching to AstraZeneca, if Pfizer was Moderna is created with the DNA copies, and in Pfizer's case, they're using E. coli, and that has problems we've just looked at with DNA contamination. AstraZeneca actually uses human host cell protein from a human cell line from abortion. It's called HEC293. That's how they make all the copies of the DNA. You need a cheap vector to make lots of copies. A bit like scaling up a factory. You want to make it as cheap as possible. You might use automation. You might put the factory in a country where wages are lower, right? People work longer hours. Pharmaceuticals do the same thing. They use cheap methods to create lots of copies to make profit. Because remember, their goal is to make money. If they don't make money, they're not going to survive in the industry. So host cell proteins are basically proteins that can be created under stress in the cell line that you're using. You could be using an animal cell line, you could be using a human one. In this case, it's human, it's from abortion, from, I think, the 1970s in Netherlands under Dr. van der Epp. And that's a number of 273, the number of 293, the number of aborted fetal experiments to get to the cell line, all products of abortion. So they tested in this study from Germany in eLife. These were experts from the University of Ulrich, and they found that of the AstraZeneca lots tested, the amount of human protein, this is human protein from the cell line, was 44 to 71% of the total protein in the shots, more than the adenovirus that was supposed to be there. So most of AstraZeneca, on a protein basis, is human protein, and it's human protein from a cell line from abortion. And how many people knew that? I know it's complicated, but let me make one point here, very important. Some things in science are complicated. They are not simple. But we as a church, we have to get to the bottom of them because vaccines are by far the only moral issue we have to contend with. Scientifically, that's complicated. Okay, we have other issues as well. Okay, IVF, chemical contraceptions, these are other examples of complicated issues that we have to get to the bottom of if we're going to have the right answer for them. So I don't make too many apologies that it is complicated. I say this shows the problem, that we have science moving into such complex areas that we can't really keep up with it. And that, I hope, shows the importance of having an event like today's. So more than 1,000 human peptides detected. And the interesting thing is, you say, well, why didn't it come up in clinical trials? Do you know why? Because they probably used an inferior technique and they got away with it. They probably used, and the paper actually talks about this, sheep and goat antibodies, and they just use that as a surrogate for human antibodies, and it doesn't work because they try to say, oh, if we inject a sheep or a goat with the vaccine, basically in a cell line, okay, will it create antibodies against the human proteins that's in the vaccine? And the problem is that actually sheep and goats are quite similar to humans for some of their proteins, okay, like cytoskeletal heat shock protein. So what happens is basically it doesn't work. Basically because humans and animals have some similarity, and therefore the animal is not going to create antibodies against itself because it would cause autoimmunity. So it doesn't work. But in this paper, they use superior techniques, mass spectrometry and other biochemical analysis. The point is, the industry is allowed to get away with it because the regulators aren't saying, hey, that technique's not sensitive enough. That's the problem. Again, it's all driven by regulators that are not strict enough. That's how you get these problems. So it's not as simple as, oh, well, it would get caught in the clinical trials. Look, those adverse reactions happened and they just took them out the trial. When it comes to testing for contamination, they just use an inferior technique and no one calls into account. That's what's happening. And then a little bit on the clots. Again, I've shared the slide before. I won't spend too long on it. But basically, there's some research from Sweden, very important paper. Here's the theory. I'll show you the practical in a moment as well. Okay, so you may know a little bit about how blood clots, basically. So I've got a little diagram there, which I'll come to the whole diagram in a moment. But basically, one of the clotting mechanisms you have is where fibrinogen, which is a protein that you have, under the action of thrombin, forms fibrin, which is a fibrin clot. So your body wants to basically stop the bleeding, so you produce a clot. And what's supposed to happen is when the clot has served its purpose and it's sealed what it needs to seal, it breaks down under the action of another protein called plasmin. Okay, so you'll be quite familiar with this, that you'll have a body that you have some kind of stemming of the tide, you can think of it as, and then it will break down eventually when it's served its purpose. The problem we have is the potential possibility for the spike protein that the vaccine is creating to act as what's called an amyloid and to interfere with natural clotting process. Okay, and that's basically what this study found. So what they did is they mixed the spike protein, which the vaccine creates, with this something that your body also has called neutrophil elastase. It's a serine protease, so it cuts up proteins. They mixed it for 24 hours, and it was producing perfect amyloid-like fibrils. And what that does, and this is my little diagram there, is when you have that natural clotting process, so you have your fibrinogen, and it's supposed to produce fibrin, and it's supposed to, under thrombin, it's supposed to break down under plasmin, and your clot goes away. Once you introduce these spike amyloids, that doesn't happen, basically. It messes up the structure, and it makes it so difficult to break down. So you have like a biophysical change in your clot structure. And that's a theory they did in the lab, and they did some analysis on it. And here's a quote, actually, from Per Hammarström from that study from Sweden, Linköping University. "We have never seen such perfect but scary fibrils as the one from the amyloid-producing SARS-CoV-2 spike protein and pieces thereof. The fibrils, starting from the full-size spike protein, branched out like limbs on a body. Amyloids don't usually branch out like that. We believe that is due to the characteristics of the spike protein." So this was the theory back in 2022, and you can see I've got a picture of one of these unusual fibrous clots that embalmers were finding, very rubbery. But now let's look at the actual practical side, when you actually study these clots. Well, recent research this year from Kevin McKen, it's a different Kevin McKen to Kevin McKernan. This is over in, I think it's in Korea or Japan. And he used, again, multiple industry standard techniques to actually study these clots that people are finding when people have died, okay? And these were actually taken from people who were alive. They didn't actually die, so they're not forming post-mortem, as some people would like to allege. he took them and he basically, well, first thing he describes, it's called the gross observations before you even get to the microscope, basically. They're rubbery, they're fibrous, they're coiled and banded, they resist tearing, they don't break down, they don't kind of just falling up to pieces. They're actually quite resistant to that. They lack the red-brown color that you'd see if they were red blood cell rich. They're not like normal blood clots like that. And he also then uses different techniques like scanning electron microscopy to basically find that if you look at this thing under a microscope and under different techniques, that it has that classic structure we looked at of amyloid fibrin architecture. So your natural clots become super resistant because of the spacing of the way that the fibrin's all packed up. It becomes like this lattice or this network that just won't break down naturally. That explains why it doesn't break down when you start touching it, okay? Because it's like a biophysical, like physics basically is what's going on here. That's why, and that would explain why it's causing so many problems for people with clotting and why the embalmers are like, I need more pressure in my pump to get this stuff out of the cadavers, right? Because it's actually a physical change, and that's what they're describing. Also, they were able to say, look, by doing elemental analysis, it's proteinaceous, carbon, nitrogen, oxygen, sulfur. It's not just heavy metals. That was one theory. It's just heavy metals being drawn from the body. It's not that. It's a natural structure. It's proteinaceous. It's a protein. And again, using morphological, histological, ultrastructural, so looking at the structure of it, spectroscopic, so looking at the graphs of it, suggests amyloidogenic remodeling of fibrin under unknown conditions. That's exactly the theory from Sweden, 2022. Now we have someone actually looking at a clot that was sent to them and finding the same stuff you'd expect. So that Swedish study, I'd say, looks dead on, unfortunately. And here's a closer look at the clots. First one, and top left, clot cut with a scalpel blade. And he makes a point in the paper saying it really does slice and break apart, but it's almost like it snaps. It's not just disintegrating. It's more like you think about something like a cucumber. It just, that's what we're dealing with. And there you can see, secondly, sample pinned to the dissection area, so it's rubbery looking. High magnification scanning electron microscopy image, top right, highlighting the ultra structure characteristic of the amyloid fibrin. It's that very, very regular, ordered, impenetrable, will not break down easily. And again, another technique using fluorescence with something called thioflavin staining to show what's called beta sheet-rich amyloid fibrils. This is indicative of it being an amyloid, okay? So he does various tests, and they say, yeah, this looks like it's an amyloid. This looks like it's causing a problem to those clots. And then, one more thing to consider, you have, this is actually established as one potential driver of, say, Alzheimer's disease, okay? Which would be amyloids. So, for example, you have certain prion proteins, and you also have amyloid beta 42, which is implicated in Alzheimer's disease, so one potential mechanism of it. Again, you can see research from Cortes, Cantelli and colleagues that fibrin blood clots, which we just looked at, in the presence of this particular protein, can cause structurally atypical blood clots, that are fibrin blood clots, and resist degradation. I've got another study there going back to 2000, Collett and colleagues. Fibrin blood clots with altered structures can be resistant to fibrinolysis, so the breaking apart under the action of plasmin. That's exactly what we just saw. So we already have other proteins in the literature where we think, yeah, actually, if you have this protein, it's a prion protein, it can cause you an issue with fibrin blood clots and they won't break down like normal. And that's exactly what they see. And you can see this protein implicated in Alzheimer's disease on the left is a normal fibrinogen clot. Look at how much more densely packed it is on the right. That's what you're talking about. It's that structural change that means it doesn't break down normally. And that's what they're seeing with the spike protein. And that's what Kevin McCairn saw in his lab as well. And then last slide on this section, something called IgG4 class switching. Now, interestingly, I showed you at the beginning of the talk a slide of Maddie de Garay, who sadly had that horrible adverse reaction, okay? That young girl was part of the adolescent trial. When they did adolescent trial, they didn't have an efficacy endpoint like they did with the adults when they said, oh, okay, you've got a 95% chance in the Pfizer trial of contracting COVID as an adult. If you take the vaccine, you're 95% less likely to have it than if you do take the vaccine. For the children, what they did is they just did something called immunobridging, and they just counted antibodies, okay? And they said if the antibody count looks high and it's good enough and it's not inferior, then basically we know it's effective because it worked for the adults. And that's how they did it. They didn't really have a proper efficacy endpoint. They just had what's called immunobridging. Antibodies are basically the golden calf or the sacred cow of vaccinology. It's all mostly based around antibodies and sometimes T cells. But the problem is there's been multiple studies that have shown that people having multiple vaccines, like 3 mRNA vaccinated individuals, are getting something called IgG4 class switching, which basically what it's doing in a nutshell is degrading the functionality of your antibodies. So the antibodies are not being as effective or as antiviral as they should be. And unvaccinated people don't experience it. People who have Moderna have higher levels than people who have Pfizer. And that's interesting because Moderna is 3 1/3 times higher dose than Pfizer. So that kind of makes sense because basically you're flooding the body with a lot of the spike protein and a lot of the vaccine. And it's basically, with this high protein amount, is leading to this class switching because you have this persistent exposure. And what this does basically on the right side is it reduces the effect of those antibodies to trigger natural processes like phagocytosis to go in with white blood cells, swallow up the pathogen, swallow up the virus. Antibody-dependent cellular cytotoxicity, so in other words, other action of other white blood cells, and also the complement system as well, which is another action of the antibodies. So yeah, you may still have lots of antibodies after 3 doses, but they're not working anymore. And that's what these studies have shown, multiple studies. Unvaccinated people, they don't show this. So again, you see that it's too simplistic of the pharmaceuticals just say, oh, we just need antibodies. Well, you need good antibodies, yeah? And you get them through natural infection, not through this technology. And then just briefly, we'll look at some views on the vaccines as well. So Francis Collins, you may know, former director of the National Institute of Health in America, led the Human Genome Project, famous evangelical Christian, convert from atheism, one of the most well-known Christians in the public sphere in the science world. He has some books at the bottom which are kind of intended to kind of get the popular message about God out there, belief, the language of God, the road to wisdom. He said, give God the glory, but roll up your sleeve. He said the COVID-19 vaccine is an answer to prayer. This is a love your neighbor moment. Prominent Christian in the science world, that's what he had to say. But the previous US government's view was a little bit more blunt. If you remember this statement from the White House Chief of Staff Jeff Zients in December 2021, we're intent on not letting Omicron disrupt work and school for the vaccinated. You've done the right thing, and we will get through this. For the unvaccinated, you're looking at a winter of severe illness and death for yourselves, your families, and the hospitals you may soon overwhelm. I've put a few famous Christians on the screen. I wonder how many of these, or leaders in the church, I should say, how many of these you think are going to be supportive or not supportive of the vaccine? Well, first up, Pope Francis. I believe that morally everyone must take the vaccine. It's the moral choice because it's about your life and the life of others. Justin Welby, Archbishop of Canterbury. Love your neighbor, get vaccinated, it's a moral issue. Ecumenical Patriarch Bartholomew I, leader of the Eastern Orthodox Church. Science, to the extent that operates as a minister of man, is a priceless gift by God. We must gratefully accept this gift. And finally, Franklin Graham, evangelical Christian. I think Jesus Christ would advocate for people using the vaccines to save lives. He's the son of Billy Graham. Now, I don't know what caused it, but in November 2021, Franklin Graham underwent heart surgery for restrictive pericarditis, November 2021, and the media reported that had started symptoms in the months prior to it. Not sure what it was, but we know that pericarditis, sadly, is a side effect of the COVID vaccines. But let's compare it to Jesus in the early church. Matthew 9:12, simple statement. I know it's not the primary point Jesus is making when he's talking, but it's still, it's actually the sub-point. But I think maybe for us, we need to think of it as the major point for this right now. Those who are well have no need of a physician, but those who are sick, Matthew 9:12. He's talking obviously about sinners. He's saying, the real bad sinners out there, yeah, they need me. If you're so perfect you don't need me, then you don't need me. Obviously, no one is in that category. But I think the sub-point is actually worth us reflecting on. Because vaccines, by the way, work as a prophylactic. You're supposed to take them when you're healthy, before you get exposed to the pathogen. It's the whole point. You're supposed to build immunity prior. That's the whole theory of vaccination. If it comes to you after you've contracted the virus, overcome it, it's not worth anything. It's an intervention for the healthy. That's the whole point. And I've given an interpretation from Clement of Alexandria in the Pedagogos, Book 1, about 198 AD. A physician is required for those of us who are diseased in body. People in health do not require a physician. They do not require him as long as they are strong. However, those who are ill need his skill. I looked through the early church. I have to be honest, they were not massively against doctors and healthcare in general. And by the way, I'm not either. And this today isn't about being against medicine or against healthcare. But they were clearly on the same page as Jesus when it comes to who needs a medical intervention. It's the sick, not the healthy. Vaccine does actually change that paradigm. It's about giving the healthy an intervention, which is different to what Jesus says. And then Just one last slide here on obligation to the state versus duty to God. Matthew 22:21. When Jesus is, they're trying to put him on the spot and say, should you be giving your taxes to Caesar, to wicked government? And you might say the same thing today. You say, well, they're doing all this terrible stuff, like abortion. Can we pay taxes? Well, Jesus says, look, render therefore unto Caesar the things which are Caesar's, and unto God the things that are God's. Look at your cash. It's an old image, Queen Elizabeth. Now maybe it's got Charles on it, the king. Who does it bear? It bears the monarch. Yeah, it's a person. The cash bears the image of a sovereign, right? That's all it is. In Northern Ireland, the cash is a bit different, right? But you get the idea. It's still got someone's face on it, some Ulster Bank president or something, right? It's just a person, yeah? It's not that big a deal. Just give your money to pay your taxes because it's just going to a person. The cash isn't made in the image of God. It's just the image of a man, isn't it? It's just creating many copies, image of the sovereign or image of the leader of a bank or something. But look at yourselves now. Look around. Man is made in the image of God. Who do you belong to? God. Who should you be honoring with the body? God. Think about it. The only commandments in the Jerusalem Council, Acts 15, what were they? They relate to the body. The only commandments that the disciples would put on the Gentiles. 3 related to food and eating. 1 relates to sexual immorality. Do not commit sexual immorality. And then 3 relating to food. No blood, no animals strangled, no animals sacrificed to idols. That's it. They were the only commands. So are we commanded to take a medical intervention as a healthy person, morally? And is that binding on our conscience? Well, if it was, shouldn't it be there in Jerusalem Council? Shouldn't it be there? Is God not outside of time? He can predict all things. We have all the prophets and all the prophetic writings. If it was something that said, and you must take the COVID vaccine, I'm pretty sure it should be in the Jerusalem Council. If it's not there, it's not in Scripture, then it's not infallible. So, in conclusion, we have seen case studies of harm from the clinical trials. We've seen 5 examples, sadly, of people, some of whom have the very discharge notes from the doctors, saying with high probability that the vaccine caused these injuries. We've seen how they've been brushed out of the newspaper headlines. They weren't covered there. We've seen how in the write-ups the pharmaceuticals do, they don't mention them. They just tell us how wonderful the technology is. But we've seen some of them have suffering from lifelong severe illness now. Some of them have been suicidal. Some of them are paralyzed. Some of them are in severe pain constantly. Secondly, we've seen a high-level review of public health trends. We've seen high-quality data from eminent researchers and statisticians. We've seen that was enough to tip it over with Vioxx, okay, to prove that it was causing cardiovascular harm. So it should be enough here because we've seen life insurance data. We've seen data from databases from Japan on cancer mortality. It should be enough. And then thirdly, we've looked at a microscopic look. So we said, okay, let's do some science. We've looked at calamari clots. We've seen the theory from Sweden in 2022 on amyloids perfectly matches the observations of Kevin McCairn's team in 2025. It looks like it's causing an issue with natural blood clot formation of fibrinogen. We've also seen contamination. We've seen DNA contamination at much higher levels. We've seen human host cell protein and the moral implications because it comes from abortion. We've also seen circulating IgG4 antibodies. Antibodies may be high, but they're not functional anymore. They're not useful. So what's the point of banging on about antibodies as an endpoint for the adolescents if they're not worth anything? And then finally, we've looked at the church, the world, and the vaccines. And we've seen that the theory of vaccinating someone who's healthy and certainly making a command out of it is not scripturally supported. It might be an idea, but there's no command that can be given to someone. No one can be compelled to do something. It's not in Scripture. And we've seen that large church leaders from all denominations, the Lord says, to whom much is given, much is required. If somebody has an office and they're responsible for a denomination of millions of people, they're responsible to get this issue right. And they should be starting with the Bible, and they should be starting with cautiously considering what has and hasn't been commanded as binding on the conscience. And certainly the COVID vaccines is not binding on the conscience. So I'd like to thank you for listening to the talk. If anyone would like any more information, my website, which is free, is alltherisk.com. So I'll put this up when it's available, and I'll put all the references up. And I'd like to thank you for listening, and enjoy the rest of your afternoon. Thank you.